right, so believe it or not, we're about a quarter of the way done with Chapter 6. There's a lot in Chapter 6, though, isn't there? Okay, so let's go to the next little bite-sized part. All right, so let's load up some Class 1 molecules. That's going to be what we are going to do. All right. First of all, we're going to be talking transporters. So the thing with Class 1 is that we're going to load the peptides on in the endoplasmic reticulum. But the peptides themselves are coming from the cytoplasm. And those are on the opposite sides of the ER membrane. And there are no holes in the ER where a little peptide can squeeze through. So you need special proteins that actually burn ATP in order to flip little peptides from one side of the membrane to the other side of the membrane. And two famous ones are going to be TAP1 and TAP2. And the TAP proteins are the ones that are going to translocate protein from the cytoplasm into the ER so that you can load it onto MHC class 1. There we go. And so these are part of a group of proteins that have a thing called an ATP binding cassette. There's a bunch of proteins that have ATP binding cassettes or ABC proteins. And the ones that we're going to focus on the most here are going to be TAP1 and TAP2, which stands for transporters associated with antigen processing. Transporters associated with antigen processing. TAP. There we go. Number 1 and number 2, and they're actually going to form a complex of one TAP1 and one TAP2, and that's going to be on the ER membrane. And their job, old immunologists would call this a flipase, because it's basically going to take a peptide and flip it from one side of the membrane into the other side of the membrane, which is harder than you might think. Yeah. Because you can't go around, you got to find a way to go through, basically. And so it also has to work for a lot of different kinds of peptide. But it can't just be, letting anything go through. So it has to be specific for peptides, but not specific enough that it can't bind to pretty much any peptide that you process. So yeah, TAP proteins. We'll learn about them. The next one you need to know is the ERAP protein. And that's the ER, endoplasmic reticulum aminopeptidase associated with antigen processing. So ERAA should probably have another A in there, and P, just like TAP, should probably have 2 A's in there, but TAP just sounds annoying, I guess. And ERAP is just too much, yeah. But it's going to be involved. I'll show you. It's going to be there helping to load things on. It is a little processor, so it's going to take a potential molecule that could fit onto MHC class 1, and it's going to trim the ends a little bit if it doesn't fit, so that it'll fit just perfectly into that MHC class 1. So you can get close with your proteasome. You can almost process a perfect peptide, and ERAP tries to clean up the little extra hanging over. It's like a little eraser. If you left a little bit too much protein on there, it just gives it a little haircut. That's all. Okay. Here is TAP, and so they're going to draw it like this. This is still, I believe, the best structure that we have for the TAP1 and 2 heterodimer. So they're just like a big chunk. You would have the membrane running right through here where I'm drawing my mouse, and just like so. So yeah, we draw them nice and schematically like this. Makes it easy to see what's going on, but yeah, that's closer to the truth. Just, I don't know, harder to recognize. They want you to be able to recognize these things by sight and color in this book. It's actually pretty good at doing that. All right, so our ABC domain, ATP binding cassette, is going to be located down here at the bottom in the cytosol. That's going to bind to an ATP and to the peptide to be transported, and then is going to break the ATP, and it is going to feed that peptide up through here. So this is the transmembrane domain. It's actually going to make a little channel that goes to the inside of the ER. So the lumen is what we call the inside of the endoplasmic reticulum, and the cytosol, the cytoplasm, that's the rest of the cell. So we are inside of the cell when we are in the lumen of something like the ER. All right, so let's keep going, and we're going to get up to the there's a nice little picture that shows all of class 1 loading in a way that I think is fairly easy to digest and test learn for later, because this is totally coming up on the final. All right. Yeah, some people are deficient, have inherited deficiencies in either TAP1 or TAP2. And remember, these two proteins work together. And so in diseases like this, they aren't always a case where you can't present. In some cases, they just decrease presentation by class 1 molecules. But yeah, surprisingly, you can survive a class 1 deficiency. You're just horribly immunocompromised, but it happens, and it's really cool that, it's not necessarily a death sentence, because, yeah, before modern medicine, it certainly would have been. Okay, other parts of the complex are going to be calnexin, and calnexin lets the next in. Yeah, you'll see it. It'll make sense in a little bit. It's like a little chaperone. It's going to bring up our newly folded MHC class 1 protein. It's going to get it ready, get it all dusted off, like that salesman in the, in the new car sales department. Shine it all up, get it ready for you. And then, the little peptide's going to come in and walk around the car, see if he likes it, and maybe lock into that driver's seat. I'll go over this again when we get to the picture. Okay. So calnexin is going to give way to this big multi-protein complex. And it's got calreticulin, it's got tapicin, and it's got ERP57. And ERAP is kind of floating around in a cloud around that, doing its thing. But these three are the peptide loading complex. So after a peptide comes through, TAP1 and 2, it is going to have to get loaded on by by these three. And I think of these as like three kids in a trench coat that are together able to reach up and just dunk a basketball through a hoop. The basketball here is the peptide, the hoop is MHC class 1, and these three are going to work together, and you watch. They're kind of all stacked up a little bit. And they are going to end up, dunking that ball. Something that I could never do, but oh well. Let's see. Calreticulin is one of those proteins, and it's in the endoplasmic reticulum. I think it is calcium binding or something like that, or calcium regulated. But it is going to be one of these chaperone proteins that's part of the peptide loading complex with tapicin and ERP57. So tapicin is associated with TAP. I think it's TAP-associated protein. Yeah, tapicin. And it's going to be, it's the long skinny one that helps to load MHC class 1 molecules. And ERP57 is going to dock onto the top of tapicin, and it's the one that's actually, it's the little kid in the trench coat that's actually going to dunk the ball through, if I remember right. Okay, peptide editing is where we check out any unstably bound peptides. So we sort of give them a little whack and see if they're going to stay in there. HLA-DM is the protein that's going to do this. And it's like, I imagine the DM is like your dear mother, sort of checking that everything's ready on your way out to school, right? Gonna, straighten up your collar, check your tie, or whatever the heck you have to wear to school these days. I don't even know anymore. It's been too long. Yeah. And let's see. Next protein is going to be Sec61. Sec61, which sounds like, I don't know, like Area 57 or something like that. Area 51, whatever it is. Yeah. Heinz 57, Area 51. There we go. And this is going to be, another one of these transmembrane pore complexes. And it's going to let peptides come from the ER into the cytoplasm. So it's like the opposite. If we have peptides that don't fit, then Sec61 is going to let him out the back door and say, it's all right. Go get processed, and when you're old enough, you can come back. Yeah. Okay, ERAD is, yes, going to be involved in specifically recognizing unfolded proteins. And this is in the ER. When you make proteins that are going to be glycosylated, that are membrane proteins, the part that's going to end up outside the cell starts out in the lumen of the ER. And there's this thing called the ERAD tuning pathway, where you go through and make sure that everything is folded properly and glycosylated properly. And if ERAD doesn't like you, then yeah, you're going to get marked for degradation. And so be it. That's a good thing. Yeah. Okay, and lastly, the retro, translocation complex. That's a heck of a word. Retro, backwards, translocation, moving things from one place to another, and complex contains more than one part. So it's a protein complex that unfolds and translocates proteins across the membrane of the ER into the cytosol. So this is going to take things out of the lumen of the ER and ship them out to the cytosol, kind of like Sec61 was doing back here. All right, so let's go. This is the picture. Isn't this great? I think this is great. So, calnexin is going to get together with the young MHC class 1 molecule. Now, this is a brand new one, was just translated yesterday, and it was translated right here in the ER. So, you just need a calnexin to kind of stabilize it for a moment. Calnexin has to be there until the beta-2 microglobulin comes in. So think of this complex as like missing a leg, and think it's just going to fall over. And so calnexin's like a crutch that keeps our MHC class 1 from falling over, you know, in my mind. Yeah. And if you want to remember it some other way, that's also just fine. Beta-2 microglobulin is then going to come in, and when it does, then we don't need calnexin anymore, and so that's going to pop off. Now we've got our peptide loading complex. And so out here in the cytoplasm, we've got some kind of ribosome. So we're translating proteins. We're making some kind of protein. Maybe it's one of these defective ribosomal products, or maybe it's going to be a normal protein. And some of each of these are going to get sent to be shredded in the proteasome. But most of the normal proteins you're going to hang on to and actually use, and all of the defective proteins you should ubiquitinate. And where are we going to ubiquitinate them? You can look back if you need to. I think it's a lysine. I think it might be lysine 48, but you go back and check your notes. And then we're going, now that this has been marked, it's actually going to bind to the 19S subunit of the proteasome. It's going to get fed into the 20S subunit of the proteasome. Proteasome is going to shred the heck out of that protein. And so we're going to get these little blobs of protein that are going to come out and are going to bind to the TAP protein. And so they're going to come through the TAP protein. The TAP protein is a translocator. And then if they just get loaded in, hey, that's great. If they're a perfect fit, no need for any extra stuff. The three proteins that are going to first sort of block off the access pathway and then open up at the right time, because, I mean, there's a lot of stuff in the ER lumen, and you don't want to load just anything on there. You want to wait until TAP has let a peptide through, then you want to open up and load the peptide in. So here's our three kids in a trench coat, tapicin, ER protein 57, and calreticulin. And calreticulin is going to take the place of calnexin. It's actually a similar protein. It's going to bind to a similar spot. But it can bind even after beta-2M is, beta-2 microglobulin is already bound. And then there's tapicin. There's a little ERP57 on top, blocking the basket, just waiting. When that peptide comes through, ERAP is going to trim off anything excess to requirements. And then, yeah, once it looks good, we're going to just dunk it right into there onto MHC class 1. And MHC class 1 will then lock down. So our peptide binding groove is going to lock down onto the beta-2M, just like so. And, yeah, we're going to export it to the cell surface. And now the cell can start presenting. So this is the process of antigen processing and presenting for class 1 MHC presentation. So there you go. This is, yeah, this is a big part. This is a test question right here. And so, yeah, focus on it. So try and write it out maybe in words. Try and draw it out in pictures with labels on everything. Try and make little arrows that show everything that's happening, because there's a lot going on here, and it's more than just what they have the arrows showing. There's a lot of little steps here. Yeah, make sure you understand this part really well. Okay, so let's just look at some of these things. Here's the tapicin. If you rotate it, it's the long skinny one. Here's our calreticulin, which is going to bind along the side. And here's our MHC molecule down at the bottom, and there's ERP57, which is the one that's going to float through and be right there, kind of attached to the top of tapicin. And so you can see here they are on MHC. They are kind of getting all around where that groove is. This is the groove on the MHC class 1 where we're going to bind things. And you can see it's kind of closed at this end, which tells you it's MHC class 1, not class 2. Okay, and I think that's a good place to pause this section, because I think you're going to want to go back and practice that MHC class 1 loading again. Draw it out on a piece of paper. That's totally what I would recommend. If we were in class, I'd pass out a piece of paper and I'd say, all right, draw it for me. Yeah. First time, use your notes. The second time, try without your notes. And then, and then once you've got it learned, then boom, it's there and you're good. All right, so thank you. That's the end of this section.